Bismillah ar-Rahman ar-Rahim. Uh, this is this lecture is about the cells of the immune system. Yeah, yesterday I have already done a presentation on the mononuclear phagocytic system. So this is generally about the cells of the immune system, and we'll see what are the different cells of the immune system, how they work, and what are their types, what are their classes. So uh, let's begin with the presentation. Okay, so the immune system, it provides the second line of defense. The first line of defense is provided by the epithelial barriers. And when we say epithelial barrier, it means where there is a epithelium present. This epithelium could be the skin, where we call it the epidermis, or this epithelium could be the lining epithelium of all the organ systems, all the systems which have a hollow cavity, which has got a space bounded by walls. It is lined with the epithelium. At certain places we call it as mucosa. So where there is some mucosal lining, for example, the GIT starting from the oral cavity, or from the respiratory system starting from the nasal cavity, then trachea, and then lungs and alveoli, and then the GIT starting from the oral cavity, esophagus and stomach and intestine, all is lined by the epithelial lining. So where there is an epithelial lining which is intact, making a continuous lining, or skin where there is continuously present epidermis, they provide a barrier. So they make the first line of defense, they make a barrier for the invasion of the microorganisms from outside into the body. The second line of defense is provided by the immune system. Once the microorganisms, the pathogens, they invade the first line of defense, they breach the first line of defense. Now comes into action immune system, which makes the second line of the defense. Now we have two kinds of immune system. One is the innate immune system, and the other one is the adaptive immune system. The innate immune system, it is non-specific. The body, since the birth, the babies, they have a innate immunity within them, which is natural immunity within them, and this natural immunity is non-specific. Whatever the organisms, A, B, C, virus, bacteria, whatever there may be, this reaction equally becomes active against all pathogens, all foreign microorganisms or foreign bodies. So this innate immune system is present, number one, since birth. Number second, this innate immune system is non-specific. It is active, equally active against all pathogens. So it is non-specific, it responds rapidly. And this innate immune system is composed of what? Is composed of the complements. Number one, the complements which are present in blood. In the blood there are many kind of proteins are present, the complement proteins. So these complements, they are part of the innate immune system. Number second, there are some peptides which are secreted from the cells of many organ systems, from the epithelial cells. These peptides are known as defensins. So the innate immune system comprised of the complements, which are peptides. Number second, defensin, which is again a peptide secreted by many cells. Number three, the innate immune system includes the macrophages and the neutrophils. When I say neutrophils, means mainly the neutrophils, but it includes all granulocytes actually. So the macrophages and all granulocytes, they are included in the innate immune system. And then there is one kind of cell which is called as the natural killer cell. So the natural killer cells, they are kind of T cells, the cytotoxic T cells. They are bigger than the normal cells, but what they have, they non-specifically kill all the tumor cells, kill all the cells which are infected with the foreign pathogens or microorganisms, virus, bacteria, parasite, whatever. So these four are the constituents of the natural immune system or innate immune system or non-specific immune system. Complements plus certain peptides like defensin plus the macrophages and the granulocytes including the neutrophils, basophils and eosinophils and the natural killer cells. They are the parts of the innate immune system. The adaptive immune system. The adaptive immune system is also called a specific immune system or acquired immune system. This system is develops after the body is exposed to some pathogens, certain pathogens. Once the body is exposed, it starts developing immunity against that. That's why it's called adaptive or acquired immune system. Now, the acquired immune system or adaptive immune system, or specific immune system, the immunity develops against that particular pathogen. That's why it's called as the specific immune system. So this is responsible for eliminating threats from specific invaders or pathogens, and they are it contains this system also includes the macrophages, which can phagocytose most of the bacteria. The adaptive immune system may react against one specific antigenic component of a pathogen. For example, let's say a bacteria, let's say E. coli. So E. coli is a bacteria. Now E. coli bacteria has a capability to activate the immune system of the body. Now, what is that part of the E. coli that activates the immunity? That part is known as the antigen. Now, not necessarily E. coli has one antigenic component. It may have many antigenic components. So the adaptive immune system reacts against the specific antigenic component of the pathogen, whatever the pathogens may be. And its ability to react against the particular component improves with subsequent confrontations. For example, at first time, when a pathogen enters, body has a weaker response. But second time, this response becomes more substantial, more strong or stronger. And with subsequent attacks, the immunity becomes further stronger and stronger. So the adaptive immune system matures with time, with the exposure. Now, what are the cells of the immune system? There are many cells included in the immune system. We have done this in the last presentation as well, but I repeat it again. These cells, the immune system cells, are the granulocytes, means the neutrophils, basophils, and eosinophils. It has lymphocytes, means the T lymphocytes and B lymphocytes both. It has got plasma cells, means the B cells, when differentiate into plasma cells, able to secrete antibodies. It includes macrophages. It also includes macrophages, which are present in everywhere in the connective tissue. Macrophages are present. So it includes the macrophages, and then it includes the antigen presenting cell. Yesterday in the presentation, I told you in detail about the antigen presenting cells, their mode of function. So antigen presenting cell like Kupffer cells, like dendritic cells, like follicular cells, like the glial cells, like the mesangial cells, all the like Langerhans cells, all the antigen present cells, they are the part of the immune system. And the one cell which just today we did not discuss about is the mast cell. So mast cell is again, it is believed that is a basophil. When it enters into the from circulation into the connective tissue, transforms into the mast cell, or the basal cell is the precursor of the mast cell. So the mast cell is a connective tissue cell. It is present within the connective tissue. Remember, it's present within the connective tissue. So the mast cell, antigen presenting cells where they are present, macrophages where they are present, plasma cells, lymphocytes, and granulocytes, all three granulocytes, they are the cells of the immune system. Okay. Now, granulocytes means neutrophils, basophil, eosinophils, natural killer cells, which are large lymphocytes. They are the part of the innate immune system. Remember, innate immune system or non-specific immune system, the cells included in the system are the granulocytes, all three. Then natural killer cells, which are kind of lymphocytes, but they are bigger cells, therefore they are known as the natural killer cells or large granular lymphocytes, plus the macrophages, plus the macrophages. These all cells, they are the part of the an innate immune system or the natural immune system or the non-specific immune system. The lymphocytes, B and T lymphocytes, the plasma cells, the antigen presenting cells, the mast cells, and macrophages. What? Lymphocytes, plasma cells, antigen presenting cells, mast cells, and macrophages. They are the cells of acquired immune system or a specific immune system. So now we know the cells of natural or innate immune system or non-specific immune system and the cells of the acquired or specific immune system. Now lymphocytes. So the lymphocytes, there are two, mainly we say that there are two kind of lymphocytes, but sometimes we do say that there are three kind of lymphocytes. And these three, the types of lymphocyte depends on their size. So the small, medium size, and large lymphocytes, right? So the small are the 6 micron meter, medium are the 12 micron meter, and large are the 18 micron meter in size. The small ones, I yesterday told you in my presentation, they are close to the size of the red blood cells. They have a small, very thin rim of the cytoplasm, and a paler nucleus. So the big nucleus is small cytoplasm. It's typically characteristic of the small lymphocytes. Lymphocytes, according to their development and timeline, the lymphocytes are classified as naive cells, means the cells which have not been yet exposed to antigens, means they have been formed into the bone marrow and they have come into the circulation and they have not yet exposed to antigens. So these cells are called naive cells. Then there are two more kind of lymphocytes, effector cells and memory cells. After the naive cells, they are exposed to antigens, they transform into, or they become mature, and now they become effector cells. Now they are able to fight to the antigens or pathogens. So effector cells, they become mature, they develop a response against the antigen, and now they call effector cells. And some of the naive cells, after they are exposed, now they become memory cells. So they memorize the exposure to antigen and they remain dormant till the next exposure. So these are the three kind of lymphocytes according to the chronology, according to the timeline. They are naive, then they are become memory and the effector cells. Functionally, depending on the function, the lymphocytes are classified into B lymphocytes and T lymphocytes. And I told you the third kind of lymphocyte is the natural killer cell, which is also known as large granular lymphocytes. But they are not exactly the lymphocytes, since they do not go to thymus for maturation, number one, and number second, they miss some of the markers. The lymphocytes in the cell membrane, they have certain kind of protein or the peptides present, and these peptides, they are known as the you have seen in your book, might you have seen cluster differentiation. They are known as the CD or the cluster differentiation subclass. You might have seen CD4 plus cells. Yesterday I told you CD4 plus cells are the T helper cells, and CD8 plus cells are cytotoxic T cells. So these CD or the cluster differentiation is actually the subclassification of lymphocytes. So these natural killer cells, and to be a lymphocyte, to be a T lymphocyte, it is must to have CD3 cluster, and to be there are three clusters to be a B lymphocytes, which come in the later part of the presentation. So these cells, they miss the CD3. Uh, uh, CD3 is peptide in the cell membranes. That's why we don't classify them into as a T lymphocytes. And they also, they have certain other uh, cluster differentiation. They miss cluster differentiation of the B lymphocytes. Therefore, we don't classify them as the B lymphocytes. But they share some of the series cluster differentiation with the other lymphocytes. That's why we call them as lymphocytes. Now, the different type of lymphocytes can be identified only on the basis of the immunohistochemical method. But yesterday in my presentation, I told you we can't classify them on the basis of the microscopic slides. We can't say that which is a B lymphocyte, which is T lymphocyte. We can only after the cluster differentiation and after the immunohistochemical reactions against antibodies, their reaction against the antibodies, we can say which lymphocyte. Now the B lymphocytes. So first I tell you the B lymphocytes, they are CD19 positive, CD20 positive, and CD22 positive. Remember, B lymphocytes, they are CD19 positive and CD20 and CD22 positive. And I told you the T lymphocytes, all T lymphocytes, they have a CD is called CD3 positive. Then we have helper and the cytotoxic T cells, this helper T cells, they are CD4 positive, and T cytotoxic T cells, they are CD8 positive. Further, so all of them, all T lymphocytes, they have CD3, but helper additionally have 4 and cytotoxic additionally have 8. Now the B cells, they differentiate within the bone marrow in humans, but in lower, in birds, they differentiate into bursa of fabricus. It is a bursa which is present close to the cloaca. So that's why they were named as the B lymphocytes. But in humans, they differentiate into B bone marrow, and then from bone marrow they migrate to lymphoid organs, secondary lymphoid organs, through the blood. And yesterday I told you from the post-capillary venules they leaked into the secondary lymphoid organs and even into the connective tissue everywhere. So into the lymphoid organs they go, and they leaked into the lymphoid organs, and there they populate, they grow in number, they undergo mitosis, they increase their number. And the B lymphocytes, they participate in humoral immunity, or humoral immunity means they produce antibodies, and these antibodies then flow along with the lymph and blood throughout the body and provide protection. Now, the B lymphocytes on their cell membrane, they have certain, what should I say, they have got receptors, and these receptors on the cell membrane, these receptors are they are known as the surface immunoglobulins, IgS, surface immunoglobulins, and mainly they are the immunoglobulin D and immunoglobulin M. So immunoglobulin D and immunoglobulin A, these are the two kinds of immunoglobulins which are present on the surface of the B lymphocyte. And what is their function? They facilitate antigen recognition. So whenever some pathogen or antigen comes into the body, enters into the body, invades the body, with the help of these immunoglobulins, they identify those and then they initiate an immune response. On the other hand, and then they present them to the T cells. So the B cells, they bind themselves to the antigen, and now this complex antigen plus the B cell bound with the antigen, it presented to the T cells. So, and yesterday I told you in my presentation, the B cell also act as the antigen presenting cells. So on the one hand, they are producing antibodies, they are taking part into the humoral immunity. On the other hand, they also be part of the antigen presenting cells. Now, after antigenic stimulation, yesterday I told you, after the antigenic stimulation, B cells develop into lymphoblast, and then they undergo mitosis called clonal expansion, and they develop into, as a result of clonal expansion, more B cells are formed, and some of them they become the memory cells, and some of them then they transform into plasma cells, and these plasma cells they start making antibodies against the same pathogen or bacteria or virus, whatever, which has attached to these B cells. So they start making antibodies against that. So if I summarize this slide, then the B cells they differentiate into bone marrow. From bone marrow they come into circulation through blood, through post-capillary venues. They leak into secondary lymphoid organs. They undergo mitosis. They increase their number. They populate the secondary lymphoid organs, and then when they are exposed to some antibody, then they become active. They undergo clonal expansion, and some of them they become memory cells, and the others they transform into plasma cells. They start making antibodies against the same pathogen which has activated them. And the B cells in their cell membrane they have sub-classification. Some of them they have CD19, CD20, and CD22 positive peptides present in their cell membrane. Now just look at this here. A single B cell yields a progeny comprising many plasma cells and memory cells. So this picture actually is summarizing what we just learned from the previous slide, that there are a lot of the B lymphocytes. When they become active they undergo clonal expansion, and as a result of them clonal expansion, some of them they become the memory cells, which become inactive. They remain dormant inside the lymph nodes and waiting for the second exposure. At the second exposure they identify the same pathogen. They start then they again undergo into clonal expansion. They make more cells. Some of them they become plasma cells, some of them again become memory cells. So now as a result of second exposure the immune response requires less time, because already memory cells are present. And then as a result of proliferation some of the cells they transform into plasma cells, and the plasma cells they make antibodies. And this antibody you can see that with the help, now this is taking place, the plasma cells are present in the connective tissue. They go into the connective tissue, they go into the lymph, they are present into the lymph nodes. So the antibody production starts either into the connective tissue or into the lymph nodes, and with the help of the lymph it circulates through the lymph into the blood, and from the blood is distributed throughout the body. Now this is an important slide. I want to tell you that what is an epitope. Now let's say this is a bacteria, and this is protein is present in the bacteria. Now this protein which is present in the bacteria you can see, so we have learned that when the antigens are for example bacteria is processed by the macrophages or antigen presenting cells. I told you yesterday that part of it is then is processed and incorporates at MHC1 and MHC2, and this MHC1 and MHC2, so we can just imagine that this is the part of the bacteria into the cell membrane. And now this bacteria is now when is presented to the T cell or to the antibody, the part of it which is recognized by the antibody or the T cell is called as the epitope. So epitope is that part of the antigen which is actually recognized by antibody, which is present in the body circulating, or by the T cells. So that is the part of the antigen which is recognized by the antibodies or the T cells is known as the epitope. So just remember that it's a small peptide, only 8 to 12 or 15 to 22 amino acid sequence, or a sugar residue which is accessible to T cells or to the antibodies. So you can see here, this is the antibody, this is the antibody. So one antigen not necessarily having one epitope, might be having many epitopes. This point may be an epitope, this point may be an epitope. So the more the epitopes, the more rapid and quick may be the response then. So you can see here, this is an antigen, it has 3 epitopes, 1, 2, 3, and these 3 epitopes simultaneously or 5 epitopes simultaneously, they are, attacked by 5, antibody molecules at the same time. I will come to this later on. Now the plasma cell, they are the bigger cells, large cells transformed from the B cells. So B cells, they become bigger and they'll develop more of endoplasmic reticulum and Golgi apparatus, because now they are involved in synthesizing the immunoglobulins. Now, yesterday I told you there's many kind of immunoglobulins: immunoglobulin A, immunoglobulin D, immunoglobulin M, immunoglobulin G, immunoglobulin E. So there are different kinds of immunoglobulins, and I will show you at the end how and when which immunoglobulin is made. Then these immunoglobulins are made in these cells, and they go to the Golgi apparatus with their PEG, and from there they are exported. Now the T lymphocytes responsible for the cellular immunity or cell mediated immunity, they originate in the bone marrow. From there they migrate to the thymus, where they become immunocompetent. Means from when they are developed into the bone marrow, when they are formed into the bone marrow, they are naive cells. From there they migrate to the thymus, and in the thymus they become mature, or they acquire the capability to function. So the immunocompetent by expressing specific molecules in their cell membrane, and for this immunocompetency they require a hormone, thymosin, which is present in the thymus. So thymosin is hormone present in the thymus. Under the influence of that, the T lymphocytes, which are coming from bone marrow into the thymus, they acquire this immunocompetency to perform their function. And what is that immunocompetency? That they express some specific molecules in their cell membrane. Now T cell surface receptors. So T, just like on the B cell, what we learned in the previous slide, that in the B cell on the surface they have immunoglobulin M and immunoglobulin D as a surface marker, as a site where they, receptor site, as a receptor to identify a pathogen, to identify an antigen. So here in the T cell, the receptors are present, which are called as the T cell receptors. So in the surface of the T cell are present T cell receptors. And T cells, they also express the cluster of differentiation, which we have done enough discussion. So CD, cluster differentiation, molecules or peptides are present, plus the T cell receptors are present. There are 3 types of T cells: naive T cells, just like B cells; memory T cells, just like memory B cells; effector T cells, just like memory B effector cells. Naive T cells leave the thymus programmed as immunologically competent, but they are not as yet ready to function. And to become ready to function, they need to be activated. So until they become activated, and when they become activated, now they undergo cell division, and then they form memory cells and effector cells. So memory cell and effector cells come from the naive cells once they become activated. And how they become activated? When they are exposed to some antigen. So the antigen, whatever the antigen, pathogen, bacteria, virus, it comes and identified by T cell receptors, attached to T cell receptors. Now they become activated. And when they become activated, they transform into memory T cell and effector T cell, just like B cells. I told you memory T cells. Now the memory T cells, there are 2 types of memory T cells again: a central memory T cells and effector memory T cells. And they are responsible for immunological memory of the acquired immune system or adaptive immune system. That the central memory T cells, they remain in the T cell rich area of the lymph nodes. So the T cell, the memory T cells, the central memory T cell, where they are present? They are, remember, they are present in the lymph nodes. They are present in the lymph nodes. Okay. And they interact with these whenever they is stimulated by antigen. So whenever the antigen presenting cells flowing from the periphery to the regional lymph nodes, these central memory T cells, they identify if they have been exposed to that pathogen, they identify that and they become active. And when they become active, means when they interact with the antigen presenting cells, now the antigen presenting cells, they secrete interleukin 12. And this interleukin 12, what does it do? It binds to the receptor for the interleukin 12 on the surface of the central memory T cells and stimulates them. So it means they are influencing one another. First, the antigen presenting cells, they interact with the central memory T cells, and they become, and this antigen presenting cell secretes interleukin 12, and when interleukin 12 comes and binds with the receptor on the membrane of the central memory T cells, now it activates them, and when it activates them, now they differentiate into effector memory T cells. So for differentiation from central to effector memory T cell, they need interleukin 12 to bind on their surface, and then the effector memory T cells migrate to the region of inflammation where they have they do their action. Okay. Now the effector T cells are of three types. So from memory T cell form the effector T cells. Now effector T cells are the three types. What are these three types? T helper cells, number one. Cytotoxic T cells, number two. And regulatory T cells, number three. So effector T cells, how many types? Three. Helper, cytotoxic, regulatory. Now remember, all T lymphocytes, I told you before that, have CD3 positive. So if a cell is not CD3 positive, we don't say it a lymphocyte. So lymphocyte, to be a T lymphocyte, it must have CD3 peptide present in the cell membrane. So it's called pan T cell marker. Pan T cell marker means CD3 positive cells. So then T helper cell 1, we divide helper cells, T helper cells into these T helper cells into 1 and type 2. And cytotoxic T cells, I told you. So T helper cell 1 and 2 means they are positive, they are CD4 positive cells. And then T cytotoxic T cells, they are the CD8 positive cells. Now, what is the difference between T helper cell type 1 and T helper cell type 2? T helper type 1 cells, when they are activated, they activate the macrophage and they inhibit the T helper cell 2. And T helper cell 2, they only activate the B cells. So remember, for the activation of the B cells, which yesterday I shown you a picture and described that picture. So the T helper cells were stimulating the cytotoxic T cell as well as the B cells. So it is the T helper type 1 cell which activates the cytotoxic T cell, and it is the T helper type 2 cell which activates the B cells. Remember, but this Th2 type also activates the eosinophils and mast cells. But remember, don't forget, B cells activated by T helper cell type 2. Well, one activates the macrophages and inhibits the T helper type 2 cells, plus it activates the cytotoxic T cells. Now, we cannot differentiate them on the basis of the slides. We can't say it's a B cell or T cell. T cells have surface receptors. They don't have immunoglobulins on their cell. And the T cells recognize, now this is a very important point, T cells recognize only epitopes present on them by other cells, means the antigen presenting cells. So antigen presenting cell coming from any part of the body, from connective tissue, any organ system to the regional lymph nodes. In the regional lymph nodes are present the T cell, or even the connective tissue, the T cells are present. And these T cells, when we say they inspect the antigen presenting cell, means they are inspecting the epitopes. So because the epitope, the part of the pathogen incorporated into the cell membrane, is antigen, and this antigen has maybe having many epitopes or one epitope, and these epitopes are recognized by the T cells. Now, and the one important point is that T cells perform their function only at short distance. They cannot, because they are present in the lymph nodes, they are present in the connective tissue, so they cannot uh, uh, travel for long distances. They close by. Their action occurs close by. On the other hand, the antibodies, they flow through with the blood, along with the blood and lymph. It goes into the circulation and it goes to the distant sites. Now, we have done this. I think we don't need this. Now, human immunodeficiency virus. You have heard about the AIDS, actually, and this was something very serious in 80s when there was no kind of treatment. But what is interesting about AIDS, that this AIDS actually attacks the T helper cells. What does it do? It binds to HIV virus, binds to CD4 molecule of AIDS, T helper cells, and injects its core, means the virus, its DNA injects into the cell. Which cell? T cell. Which T cell? T helper cell. So the virus, number one, binds to the molecule. Which molecule? CD4 molecule of a T cell. Which T cell? T helper cell. And then injects its DNA inside the cell. Then virus incapacitates the cell. That's how it incapacitates a T helper cell. And now you know that from this discussion, now we know that it is a T helper cell actually which initiates the immune response. It stimulates the cytotoxic T cell as well as the B cell. So now these T helper cells, they become incapacitated, they become incapable of initiating any response. Why? Because the virus of the AIDS or the immunodeficiency virus actually binds to the CD4 plus molecule and injects its DNA inside the cell, and so the more and more viruses are made and incorporates into the CD4 molecules. So they become incapacitated, they become they become incapable of, or they become inactive. They cannot be activated now. So the CD4 plus cells or T helper cells are attacked by the AIDS virus. Now this picture gives an idea about cell mediated immune response, that this is an antigen presenting cell, a macrophage, infected cell, eating infected cell, processing it, and when it processes it, you know that it incorporates it into its own cell membrane. So you can see now these are the epitopes present in the antigenic cell pathogen, these spots, red spots. Now you can see these red spots have been incorporated into the membrane of this at MHC2 or MHC1, both locations, and then it's presented to a memory T cell and that flows along with the lymph to the lymph node, presented to a memory T cell, and now this memory T cell undergoes clonal expansion. Some of these cells, they become memory T cells, or some of them they become effector cells, and then the effector cells, they attack the infected cells, and simultaneously the natural killer cells also they attack which are the part of the innate or the non-specific immunity, also attack. They release the gamma interferon and the tumor necrotic factor alpha beta, and they attack these infected cells so that the cell becomes dead. So this is the cell mediated immunity. Cell mediated immunity occurs with the help of two cells, cytotoxic T cell and natural killer cell. Remember, cytotoxic T cell through antigen presentation, and natural killer cell directly without any antigen presentation. These are the cells which directly attack the infected or the tumor cells or the infected cells. The same which I told you, This is another presentation. CD4 plus cells means the T helper cells. We say two kind of T helper cell, T helper cell 1. Sometimes they say three kind. They also say T helper cell 0. So T helper cell 0 is that cell which actually undergoes mitosis and makes T helper 1 and T helper 2 cells. So the CD4 positive cells, T helper cell 1, when it activates macrophage process antigen present to T helper cell 1, it becomes activated, and when it becomes activated, it activates the macrophage again. It's yesterday in my slide I shown you this um, flip kind of figure where arrows was leading to that. So I told you that the T cells, they stimulate the macrophage and macrophage stimulates the T cell, and then T helper cell 2, stimulates the uh, B cells, and then the B cells synthesize they transform into the plasma cell, and plasma cell synthesize the antibodies. And here the cytotoxic T cells, they are CD8 positive cells, and they directly, after they are stimulated from T helper cell type 1, they attack the pathogens directly, and then natural killer cell or large granular lymphocyte, it's CD16 positive. So you have to remember, maybe in the paper or in the viva you may be asked these cluster differentiation. So the natural killer cells, large granular lymphocyte, CD16 positive cells, the cytotoxic T cells, CD8 positive cells, and T helper cells are the CD4 positive cells. This is the antigen presenting mechanism. We have done it yesterday. You can just read it. We have done it. This is about the natural killer cell. They are CD16 positive cells. If you remember that, here in this picture, CD16 positive cells, natural killer cell, or large granular lymphocytes. Why we don't call them? I mean, we classify them as lymphocytes because they look like lymphocytes, but actually they miss CD3, which is a pan cluster differentiation. They don't have CD4, CD8, which is the cytotoxic, and the T helper cell cluster differentiation. They're only CD16 positive cells. So they recognize and kill virus infected cells and certain tumor cells. Some of them, they may perform antibody dependent cellular cytotoxicity. Means when a cell, when a pathogen is coated by antibody, antibody attaches to the pathogen, or a cell is attached by an antibody, then they also, the natural killer cells or the LGLs, they also attack these antibody coated cells. So that's why they call the antibody dependent cellular cytotoxicity. They secrete, natural killer cells, they secrete interferon and interleukin 1. So this is important. So remember that they secrete interferon and interleukin 1. Thus, this slide is summarizing what we have done. So you can go through that. Bone marrow B cell, stem cell synthesizes both T cell and B cell. B cell comes into the circulation. This is naive B cell, and then within the lymph node, in the tissue, in the lymph node, this B cell, they when exposed, activate by some pathogen, transforms into the plasma cell. Some of them, they become the memory cells. The T cells, they become T helper 1, T helper 2 cells, or cytotoxic T cells, and from their circulation into the thymus, and from the thymus through the circulation they come into the lymph nodes and into the connective tissue. Similarly, the natural killer cell arising from the same cell, and from here it comes into the tissues and into the circulation. This is in detail we have done yesterday. It's the repetition of that. How the antigen presenting cells, they function. I told you, you can go through it, you can read it. Monocyte macrophage system we have done in detail. Now the mast cells, the last cell, the mast cell. The mast cells, they are very common cells. They are present in the connective tissue. If you remember the cells of the connective tissue, so mast cell is one of the cells of the connective tissue. It's present, skin, GIT, serous membranes. They are particularly numerous around small blood vessels. Remember, this is a very important location. They are numerous around small blood vessels. Functionally, they are just like basophils. Like basophils, they are characterized by the metachromatic cytoplasmic granules. In their cytoplasm, they have got granules which are metachromatic, means we change their color when they are stained. And what they contain, just like basophils, they contain histamine, heparin, and serotonin in their cytoplasm. And it's believed that they usually, actually, the mast cells, they have come into the connective tissue from the basophils. Now, how they function? When they function means they degranulate, means they release their granules. When we say they release their granules, means what? Means they release histamine, they release heparin, and they release serotonin. So by the exocytosis, they degranulate, they release their granules, and from the granules, histamine, heparin, and serotonin is released. Now, when they degranulate, for their degranulation means the mast cell, inside the mast cell, granules, and granules to come out of the cell into the connective tissue. How and when? When the mast cells are stimulated by immunoglobulin E. So until and unless mast cells are stimulated by immunoglobulin E, they will not degranulate. They will not release their granules, means they will not release histamine, heparin, and serotonin. And when they release that, and by activation from immunoglobulin A, that is called hypersensitivity type 1 reaction. You sometimes you also call it allergic reaction. So acute allergic reaction, sometimes you know that somebody eats some peanut and immediately develops an immune response with choking throat and flushing of the face, dropping of the blood pressure, fainting. All that is because of this hypersensitive type 1 reaction, where I told you where they are present. They are present in numerous quantity around the small blood vessels. So the immunoglobulin E, comes to these mast cells, attaches to the mast cells and degranulates, and histamine is released, causes vasodilatation, heparin and serotonin. All these are the mediators of the hypersensitive type 1 reaction. Now let me go to the 2 slides which I left earlier, and I tell you what are those slides. Now the same plasma cell synthesizes all immunoglobulins. How many type of immunoglobulins do we have? We have, how many types we have? We have the immunoglobulin M, immunoglobulin G, immunoglobulin A, immunoglobulin D, immunoglobulin E. All these are the types of antigens, antibodies, or immunoglobulins. Now see, important, and remember this. When a naive B cell, means a naive cell, is activated, so first antibody which makes is immunoglobulin M. And this immunoglobulin M, when bound to the surface of an invading pathogen, it activates the complement system. So the first B cell is stimulated, and when stimulated, transform into the plasma cell. And this plasma cell, the first antibody which it makes is the immunoglobulin M. This antibody M, or immunoglobulin M, then goes and combines with the complements in the circulatory system. And this immunoglobulin M molecule, so when it binds to the complement, means it also initiates an innate immune response. It also activates the natural immune response or non-specific immune response. On second, this immunoglobulin M molecule also binds directly to the virus, so that they cannot contact to the cell surface, so protecting the cell from the viral invasion. So when the immunoglobulin M binds to the foreign pathogen, they are not able to invade the cells, rather the antibody dependent cytotoxic cells, they start killing the virus bound with the immunoglobulin M. Now, once the immunoglobulin M is produced, the B cells now they can manufacture to make another kind of antibody or immunoglobulin. Now, this mechanism or this phenomena is known as class switching or isotype switching. So first immunoglobulin which is made, immunoglobulin M. Once it is made, now they can make immunoglobulin G, immunoglobulin A, immunoglobulin E. Now, when they make which immunoglobulin, let's read it. Now, this depends on the particular cytokines that are present in the B cells microenvironment. For example, and these cytokines are coming from where? These cytokines are coming from macrophages. These cytokines are coming from T cells. So these cytokines which are coming from macrophages and T cells, depending on that cytokine, now the B cell, means the plasma cell, will synthesize what kind of antibody. So let's see how. These cytokines are released by T helper cells and as a function of the type of pathogen present. So now let's see what. During a parasitic or worm invasion, if a worm or a parasite has invaded, the T helper cell release interleukin 4 and interleukin 5. Important. So if a parasitic infection is there, let's say in gut some parasites are present, in the connective tissue, the T helper cells will release interleukin 4 and 5. And the B cells now they differentiate into plasma cells and they release what? Immunoglobulin E. And this immunoglobulin E, now what does it do? It binds to the mast cells, degranulates them on the surface of parasite. So the parasites are killed now. But during bacterial and viral invasion, the T helper cells they release interferon gamma and interleukin 6, interleukin 4 and 5 during parasitic infection, interleukin 6 and gamma interferon during bacterial and viral infection. And now the B cells transform into plasma cells and they make immunoglobulin G. Here they were making immunoglobulin E. Now they are making immunoglobulin G to fix the complement number 1 and stimulate natural killer cell, what I told you just before, to kill virally altered cells directly. This is called antibody dependent cell mediated cytotoxicity. I told you that. And then during viral bacterial invasion on the mucosal surface, T helper cells they release what? They release tumor growth factor beta, and now the B cell make immunoglobulin A so that the immunoglobulin A is released on the mucosal surface. So immunoglobulin A is released on the surface of the GIT, on the surface of the respiratory system, on the mucosal surface. So depending on the cytokine which is released from T helper cell. This is very important point. So T helper cell releasing the beta tumor growth factor beta, the B cells transform into plasma cells which release immunoglobulin A, and this immunoglobulin A then along with the secretion of the gland, it will come onto the surface. If the T helper cells are secreting gamma interferon, here it was tumor growth factor beta and beta, and here it is gamma interferon and interleukin 6. The B cells will synthesize what? Immunoglobulin G. If they are secreting interleukin 4 and 5, they will synthesize immunoglobulin E. So you remember this. So this is same plasma cells, depending on what kind of cytokine, what kind of interleukin coming from the T helper cells, synthesizing different kinds of immunoglobulins. I'm sure this presentation of mine has helped you in understanding the concept of immunity, what is natural immunity, what is innate immunity, I want to show you one more. Let me go to this picture one. The first picture, which you saw the first, yes, this picture. Now look at this picture. These are the sites of the which are providing the first line of defense. This picture. For example, in the tears of the eye, an enzyme is present, lysozyme, which is bactericidal, protects against invading bacteria. Similarly, the saliva coming into the oral cavity, which washes away the bacteria. Similarly, the nasal cavity has got kind of filtering mechanism and immunoglobulins coming onto the surface, which are protecting against the invading organisms. And there are mucus is present, and there's ciliary mechanism which washes away the bacteria, traps the bacteria. Similarly, in the bronchi we have mucus, and in the bronchi, within the lining of the trachea has cilia present, which are moving away, washing away the microorganisms. Similarly, in the gut, we have got the acidic environment which kills the bacteria. Similarly, skin has an acidic pH, plus it provides a intact epithelial lining is present, so it's actually impeding the bacterial invasion. Similarly, in the small and large intestine, we have got some kind of cells, immune cells present, called M cells, which are antigen presenting cells. Plus, the lining, there are some cells which are secreting defensine, which I told you in earlier. So this defensine comes onto the surface and protects against the bacteria. And similarly, the low pH, acidic pH of the vagina protects against the invading microorganisms. So all these mechanisms, the intact epithelial lining, plus these secretions, and plus these mechanisms in the nasal and respiratory system, and this pH mechanism, all they are act as the first line of defense. So this is the first line of defense, and immune system is the second line of defense. And the natural immune system or innate immune system is the second line of defense, and adaptive immune system or specific immune system is the third line of defense. I hope this has cleared your much concepts about the cells of the immune system and concept of the immunity. If you have any question, you can direct me. You can ask me. You can find my email address. You can write to me as masood.ahmed@frpmc.edu.pk. masood.ahmed@frpmc.edu.pk. You can also write your questions to me on the Edmodo in the module at the site of the module. Thank you very much. पता नहीं रिकॉर्ड हुआ या नहीं